we have Talmo Pereira speaking to us today. So I'm excited. I actually worked with Talmo last summer at Google, and he told me a lot about his work on using post-tracking software. And so it seemed like that would be a cool tutorial to present to the department. So Talmo is a PhD candidate at neuroscience at Princeton University. And so today he'll be talking about his work using deep learning to perform post-tracking.
Thanks, Janelle. All right, guys. Excited to be virtually here. So today we're going to be doing this in two parts. First, I'll be giving a little somewhat long talk on how we've been using animal pose tracking to better understand behavior. And afterwards, we'll do about 30 minutes of a tutorial on using the software that we've developed to do a multi-animal pose tracking. So as we go along, there will be several, I think, natural stopping points between sections if you guys want to stop and ask questions. And of course, the tutorial afterward will be a lot more interactive. So let's get into it. So behavior. Behavior, particularly through the lens of neuroscience, is typically schematized in something that kind of looks like this, right? Where we have a body that controls effectors in response to sensors to produce behavior. Put more biologically speaking, we can consider this as being the brain, which through the spinal cord or equivalents controls muscles to create behavior to interact with the physical world in response from signals in the environment. In other terms, behavior then could be considered akin to body movement. And this is going to be the operant definition that we're operating on here. So our goal with the methods I'll be describing today is to go from videos of behaving animals, that is moving animals, in this case we have here a fruit fly, to some quantitative measure of movement. So how are we going to go about doing this? All right, so first we're going to start with a short background on animal tracking using computer vision. That's going to motivate us for how we started developing our software called LEAP, in which we first adapted deep learning for animal pose estimation. Then we'll get into the new version in the successor of LEAP called SLEAP, or social LEAP, which enables very general purpose animal pose tracking. And we'll get into the challenges that emerged as we evolve from one to the other. And then finally, I was going to end with a couple of stories of how we can use unsupervised machine learning to recognize behavior directly from pose tracking or animal body movement signals. Cool. Yeah. So background, how do we go about tracking animals, right? In the course's description, and this is something that you might not have even formalized, but certainly you've seen in the past, is starting from these course descriptions, either of a centroid or the position of the animal to an ellipse. And that might look a little bit like this if we overlay it on a mouse. In an ellipse, we will fit a centroid and we'll be able to derive its direction. The next level of description allows us to do this with multiple animals. This introduces a variety of challenges, particularly pertaining to how we keep track of individual animals across time. To then the finer and description of behavior, which is now all of the rage, particularly in neuroscience and behavioral neuroscience, that is to do pose estimation, in which we go from having a single point or a pair of points to many landmarks around the body of the animal. And the final frontier, as we'll hope to get to soon as collectively as a field, is to do 3D pose estimation. So I'm not going to be talking about this in this current talk, but there's lots of exciting directions in this at this finest level of description. So why is it not enough to do the classical tracking, to do center and orientation? Well, consider these three different fruit fly behaviors. For something like walking or general locomotion, this is where you traditionally would use the center of mass, and as you can see from this tracking here, you can indeed extract a signal that corresponds to these dynamics. You see the movement of the center of mass. And perhaps the orientation gave you a little bit more information. But for other movements that do not involve locomotion, like say a fruit fly grooming. Here you see its two front limbs rubbing its head. There is no change in any of the parameters that we can capture with classical tracking, nor for this behavior called wing extension. So we're missing information even in this case about the specifics of the gait, and in these cases for the limbs that are simply not captured in this coarser description of behavior. So this is where pose estimation comes in. The idea is to extract the position of every body part such that we can now capture dynamics or motion of every actuatable limb of the animal, and therefore have a complete description of all the types of behaviors that its motor system can elicit. So how are we gonna go about doing this? So for this, we developed a system called LEAP. LEAP builds on top of previous work in human pose estimation using deep learning, which has had tremendous success in taking images that are completely unconstrained, running them through a convolutional neural network that then is trained to predict these heat maps. So these are images in which the brightest pixels, like these red ones here, correspond to the likelihood of the body part being at that location. So this totally changed the game in the human pose estimation field, in particular when they came up with this representation, which is an image rather than the raw coordinates. So doing this makes it, it's very amenable to neural network training, particularly in these fully convolutional networks. And so this is the approach that we decided to adopt. But we had a problem in attempting to do this with animals. That is, how do we deal with having no training data, right? So although these approaches in humans were wildly successful, they relied on having these training data sets on the order of tens of thousands to hundreds of thousands of images. And that's simply not tractable to annotate and gather for lab animals, particularly when you have a new animal, new morphology, or new imaging condition. So we developed to deal with this, we developed a software framework called LEAP. LEAP stands for LEAP Estimates Animal Pose, in which we have this general pipeline where we go from raw images of our animals into a convolutional neural network that then is going to output these part confidence maps. So these are these probabilistic representations. Then we'll generate the pose. So getting into the details here, the idea was to create a simple and lightweight encoder-decoder style convolutional neural network. And what that means basically is that we start with the raw image of our animal. That's going to be fed into a series of convolutions with pooling. This enables us to capture features across different scales in the image, and then upsample it until we can now output a multi-channel image where each channel corresponds to the part confidence maps for each individual body part. And then we can just train the network based on the ground truth body parts that we generate from labeled data, such that it can now do this on unlabeled images. Great. So we implemented it, and it's a tiny network. So the first thing that we noticed that we were able to achieve was very fast training on a single GPU. So during the training phase, in as few as 15 to 20 minutes on a single desktop GPU, you can already get to virtually human level or convergent accuracy. And so here on the right, what you have is a visualization of the accuracy distribution, that's these circles here, over multiple training epochs. And as you can see, by 15 epochs or so, and that's about 15 minutes on just a desktop GPU, single one, rather than on a huge GPU cluster, you can already get to virtually perfect accuracy. So because we could do this fast training, this enabled a paradigm called human-in-the-loop training. And this is the key to being able to adapt this to new data sets of new animals with new sets of body parts. And so the way that works is that we have a GUI in which you load up your images for labeling. You would click and drag the markers for the body parts that you've defined, such that they're positioned on top of the correct locations. You'd label as few as something like 10 frames, train just for a few minutes, then the software would predict on the unlabeled frames, which then would get imported back in, and all you'd have to do is correct those labels. And as you saw from the visualization in the previous slide, very quickly we can get to a pretty reasonable approximate accuracy, such that as we continue going through this loop of labeling and training and estimating and then correcting, the amount of time that it takes to generate a new training sample to label a new image decreases drastically. Because now, as we increase the number of labeled frames, all we have to do is fix fewer and fewer mistakes. So this means that basically you can get to a full data set of maybe 1,000 images or more effectively in an afternoon in a sitting. So something that's a very tractable time scale for experimentalists, for lab scientists. Further, we found that even with very few labels, on the order of hundreds, you get to the plateau of how accurate you'll be. So put together, this means that this whole system can start completely from scratch and get you to a fully labeled data set that gets you, accuracy that you can actually do work with within a very short amount of time with very little labor required. In fact, when we finish creating our data set, we train it with our full set of training images, we get incredibly accurate prediction localization errors. So where you can see here, we have errors on the order of microns relative to the size of a fruit fly, which in image space is only a couple of pixels, even in the 90th or the highest percentiles. So what does that allow us to do? Well, for behaviors like locomotion, which previously we could have used classical methods to perhaps track the centroid, now we can use this to actually quantify the gait dynamics. So gait stride and how each leg is employed separately. And later I'll show you how we use this to dig into how locomotion works at a finer resolution than we could before. But this also allows us to capture behaviors like grooming, which previously we couldn't measure very easily, because now we can track the positions of these limbs, even when they're closely interacting with other body parts. Great, so here you can see the resulting signal. And this is the kind of data that we want to be able to quantify the more interesting behaviors. You can see that we can capture the cyclical structure, for instance, of this grooming bounce. So we designed this for fruit flies because I work in a fruit fly lab, but what we found quickly was that this actually generalizes very easily to a number of different animals. And if this video playback is choppy, you can check out the presentation link from the GitHub or this URL shortener to check out the full resolution videos. But as you can see, we and other people who have used their software have applied it to a variety of animals, including giraffes, ants, moths, and of course mice, as well as more eclectic set of animal body type morphologies like hydra, or here, this was part of our original LEAP tutorial, a depth camera imaged cat chasing a laser pointer, or just downloaded from Twitter, from social media, a cat chasing a ball. Right, so this is all to illustrate that it can handle really a wide diversity of image conditions. It's really not limited to particularly the lab setting or to more controlled image settings. So great, we did that, and we could track single animals, but there are several other challenges that LEAP didn't quite handle, and in particular, dealing with multiple animals. So that led us to design the successor of LEAP, called SLEAP. Oh, sorry, maybe I should stop here for questions before I go on.
Yeah, it looks like there's a couple of questions in the chat.
Yeah.
So we can you can read them off, or
Great. I can do that.
Yep.
So how many FPS on what GPU? So when we did our original benchmarking, we were able to get 185 frames per second on 1080 GTX Ti. However, especially you'll see this especially in SLEAP, we're able to achieve incredibly higher frame rates by reducing the resolution either of the input or the output. And there's some tricks that have to do with that and balancing that with accuracy. But effectively, if you can deal with like coarse enough accuracy, you can go up to like thousands of frames per second, completely enabling like real-time prediction. So the requirements in terms of image, video, and spatial temporal resolution, that is going to be a complete continuous parameter. So the rule of thumb is if you can see it and reliably click on the body parts, then you should have sufficient spatial resolution to be able to reliably locate those body parts. If the body parts are very blurry, this is probably a result of having a low temporal resolution. So that's when you might want to increase the frame rate of your camera. And you're going to notice this again while you're doing your labeling. But there's no hard limits. So do you have to fix the labels on frames one by one, or once one is fixed, the rest follow? So you'll fix the labels on a subset of the frames, and you can at any point stop fixing the labels, retrain, and repredict. Have you tried this with insects like caterpillars, which appear to lengthen or shorten as they crawl and are able to raise their bodies or climb vertically? Yeah, so I'll bring back this slide here. So not quite caterpillars, but we have done this with worms that have similar, I guess, image feature appearance. And things like lengthening and shortening, as you can see with this hydra, for example, aren't a terrible issue. And part of dealing with that is in having architectures that can integrate information across scales. In terms of going in and out of the perspective of the frame, that is like climbing vertically, for example. So you can see in this example here where the cat is coming from further from the camera towards the camera. And it's one situation in which you can handle that. But here in the mouse example, perhaps you can see that a little bit more saliently, in which you can see what happens when the mouse rears even away from the camera, right? So what happens is you can mark key points as being missing altogether, in which case the network will learn to predict that points are occluded, or you can force it to predict where you think the body parts are. It's all about how you choose to label. So, all right, LEAP was pretty cool. It estimates animal poses, or rather animal pose. But we're in a fruit fly courtship lab, and part of our goal was to actually develop these algorithms so that we could track the movements of multiple animals while they're socially interacting. And that introduces a set of different issues that pertain to the problem of having multiple animals. Right, so Rebecca just asked in chat about dealing with blobby key points. There's certainly a lot of discussion having to do with that, and it's a big issue. You can see that with mice, for example, we typically have that problem because it's hard to see key points, particularly from above. But let's discuss that afterward. There's lots that you can do in that domain. All right, so then we developed SLEAP with several goals. First, to have a more sophisticated GUI, have it be all in Python, and have it be easy to install. As if you've worked with any of the deep learning-based frameworks, you might be familiar with the headache that is installing CUDA and having GPU support. So it's been a long time in development, and we now have it publicly available at sleep.ai. And this is going to be the software we're going to be working with, and we're going to go through the tutorial in just a little bit. So, in particular, SLEAP was developed to deal with two main problems. One is, how do we deal with the problem of having multiple instances? And by instance, I mean multiple copies, if you will, of each animal. And the second is, how do we adapt to different data sets? So although I showed you a variety of different data sets in which we're able to apply LEAP, it still doesn't work as well as we'd like it to across the board for different data sets with different kinds of image properties, and we'll look at those in a moment. So first, the problem of dealing with multiple instances. So this is actually there's a set of very principled approaches that come from classical computer vision, in which they kind of break down into either a top-down approach, in which basically you begin by predicting something about like a high-level description of your system, in this case our animals, to in some sense simulating what would happen if we isolated the instances. That is, if they were centered and cropped to then analyzing those simulated images. And what that's going to look like is that we're going to crop the images of our animals. And in our alternative approach, we have this bottom-up technique. And just one note, we have a little bottom-up problem. All right, a little animal problem right here that's seeking to deal with the bottom-up approach by chewing through my computer cables. All right, but in classical computer vision, this technique involves first analyzing features in the image. So in our case, that's going to entail finding the body parts of the animal, and then parsing them using some contextual information into a full prediction of the pose of the animal. So going from low to high level. And so how do we implement these two approaches in SLEAP? So first, the top down. We start by isolating the instances, and we do this by finding an anchor point on the animals. So usually like either the center of mass, the centroid, or a very easily visible body part. We are going to then crop centered bounding boxes around our animals, such that we're simulating what would have happened if the camera were following the animal, and we're always centered around that particular body part. And then we're going to basically perform single instance part detection. So just like we did with the original LEAP, we're going to do it such that we're going to predict the locations of the body parts using these confidence maps, but only for this centered animal. And so the key thing here is that the network learns to deal with multiple animals by leveraging the fact that the focal animal is the one that's in the center of the crop. And in that way, it can learn to ignore detections of the same body parts of animals that are in the surrounding. And great, this works remarkably well. This is something that can be easily adapted for a variety of approaches, including the other existing single animal pose trackers. So what about the bottom-up approach? So this actually deals with this completely differently. In the bottom-up approach, we first start by training a network that can predict these confidence maps for all of the instances. So we're going to take in, rather than a crop, we're going to take in the entire image. And within this image, we're going to have a network that can predict the location of every body part of every animal. And we can convert these confidence maps back into coordinates by doing local rather than global peak detection. And great. So you say, well, maybe now I have all the body parts for all of the animals, and maybe I'm done. But you have to deal subsequently with the grouping problem. And the grouping problem entails resolving situations like this, where say that you have two copies, two instances of the same body part, like say the thorax of a fruit fly and the tip of the right mid-leg of a fruit fly. And you have these two. You have two different hypotheses for how they might be grouped together. One in which this thorax is grouped with this leg, and this thorax is grouped with that leg, versus the actual correct hypothesis of grouping this body part with this thorax with this leg, and this thorax with that leg. And there is no information in the images alone that allows us to disambiguate between these two hypotheses. So what we're going to do then is we're going to borrow another technique from computer vision called part affinity fields. And in this technique, the idea is to represent the connectivity between body parts as a set of unit vectors that point along the direction from the source body part to the destination body part. So this is something that, again, we're going to define from the ground truth data. And all you need to do, in addition to having the locations of the body parts, is you need to define a directed graph that spans all of the body parts. So this is defined once in the humans, but obviously you'll have to define it differently for each type of animal. But once you do, you can leverage these part affinity fields, which incidentally are predicted by a neural network, to then parse out the body parts into which animals they belong to. And so if we take a line integral, that is, if we take the average of all of those directed arrows from each pair of candidates, then we can weigh these arrows here, or the strength of the connection, by how much the arrow in space aligns with the arrows that were predicted in the part affinity fields. We're going to do this for every connection that forms the body skeleton graph. If we do this for every connection, and I'm showing you here all the possible pairwise candidates, it might look a little bit like this. And what you might be able to see if you start squinting is that there's some clearly some very weak connections and some very strong ones. So with a greedy matching algorithm, under the assumption that this is a directed tree, we can actually find an globally optimal solution using greedy matching locally. So by resolving all of those errors, we'll get something that looks like this. So the output of our neural networks, so you can kind of visualize that over time, looks like this for our part affinity fields. So this is akin to our confidence maps that we saw previously. And this is what it looks like after parsing. So you can see that even when they're closely interacting, we're still able to resolve those body parts. In fact, even when they're completely overlapping. So the network is able to handle this problem of inferring these part affinity fields quite well. And so summarizing this and putting this in the context of how we do single instance pose estimation. In a single instance case, we have an image that goes through neural network to generate our confidence maps that have a single peak, in which we can do global peak detection for each of these different colors, which belong to different image channels. Then in the top-down approach, as we mentioned, we start with a full frame. We do a region proposal, that is, we find some sort of anchor from which we can crop the instances, and then we feed those through a second neural network that predicts the locations for the individual instances. Whereas in the bottom-up approach, we have a single neural network as opposed to two that takes a full frame and simultaneously predicts both the confidence maps and the part affinity fields, which allows us to then do our part grouping through our graph parsing algorithm. Great. So that's wonderful, and we'll talk a little bit about the pros and cons of each of these methods, but clearly you can already see that in the top-down approach, we have to deal with two separate branches of the network, or two networks entirely, separate networks entirely, whereas in the bottom-up approach, we only have to do this once. And the second biggest difference being that this, the top-down method will scale with the number of instances that you have. That is, you're gonna have to run each crop through the second network, such that for each animal that you have, you're gonna incur like a linearly increasing runtime cost. So before I go on to problem 2, I'll address the one question that we have in the chat. How do you deal with occlusions, especially when one animal is entirely bisected by another animal, as sometimes occurs with mice? And 2, when a key point is totally occluded, you get missing data. How do you typically deal with this in downstream analysis? Okay, so for the first part, we deal with occlusions in a couple of different ways. And in the top-down method, this is actually kind of dealt with intrinsically, right? Because we can choose an anchor part, and as long as that part, the crop is anchored and is centered on that anchor, the network can implicitly learn to bridge those gaps. But this will be a problem for techniques like the part affinity fields, in which it depends on how you define your skeleton tree. But we can talk about that a little bit more later. And when a key point is totally occluded, you get missing data. And this is something that you can actually just really mark in as part of the labeling procedure, such that the network can actually tell you that it does not think the body part is present. We deal with it in a variety of different ways, and it depends on the kind of analyses that you're doing. But a simple way to deal with it is to impute the missing locations through a simple interpolation, or through, for example, state space filtering models like a Kalman filter, things that can predict, given context, where those body parts are. For the most part, we attempt to be completely agnostic to this as far as SLEAP goes, and leave that decision down to the user for whatever is more appropriate for their downstream task. And we can totally talk about that also later in the tutorial. All right, so getting back into the problems we dealt with in SLEAP. So the second problem was adapting to different data sets. And so first things first, we found that, even with multi-instance data sets, we're still able to retain our sample efficiency. So what that means is that here, what we have plotted is on the y-axis accuracy. This is an accuracy metric. We talk about if you guys are curious, but it's just an overall accuracy metric, more suited for multi-instance prediction. And what we find is that as we increase the number of labeled frames, we're able to still get to a plateau of accuracy fairly quickly. Great. So we went ahead and started applying this to different data sets. And what we found was that depending on what animal that you're trying to track in the particular setting, you're going to deal with very different image features and properties about distributions of the animals themselves that are going to lead to the use of the two different kinds of approaches, as well as adapting your neural networks to better deal with the different cases. So we can have situations where we have very large instances like these mice that occupy a large part of the image, but have a small field of view, and yet have coarse features. So this goes back to the blobby type problem that Rebecca mentioned earlier. So where we might have less certainty about where the body parts might actually be located, versus a data set in which we have very small instances, but a large field of view, so a lot of background pixels to deal with, and very fine features. So in this case, we want to have as much resolution as possible, but we want to be able to ignore the background as much as possible as well. And contrast, and then the hardest case being where we have very large instances, very large fields of view, and yet still very fine features like these antennae and these beaks. So how are we going to go about dealing with all these different scenarios? Well, we started to look at specifically the neural networks that were dealing with the different problems, with the task of predicting the different representations that we talked about, like confidence maps and part-affinity fields. And we abstracted that away into this general template of encoder-decoder convolutional networks. And so in this general architecture design, we start with the input image, followed by a series of encoding steps. So these extract features across larger and larger scales to then upsample it back to produce your output through your decoder. And specifically, the blocks that are contained in here are convolutional blocks. So these, as we increase the number of filters or parameters, we increase the representational capacity of our network. That's one easy knob for us to turn. But the number of downsampling blocks is one of the key things that we're going to be playing with. And so these are able to increase the receptive field size, or how large an area of the image that the network is able to reason about. And the upsampling blocks will then enable us to recover spatial resolution, so you can get finer features back at the end. And skip connections allow you to fuse features across scales. And so, as a point of reference, here's 3 common architectures that you may or not be familiar with. Like the LEAP architecture effectively looked a little bit like this, where we had no skip connections, but a symmetric encoder and decoder. The Deep Lab cut, if you're familiar with that, I don't know who isn't to this point, uses a ResNet or MobileNet, another pre-trained network, as the encoder to extract all these features and generate a small resolution but deep feature bank to then upsample it back to the output. And UNet, a very popular architecture in image segmentation and biomedical applications, uses a set of repeated pooling and upsampling steps with skip connections. So this is effectively just like the LEAP architecture, but with skip connections, which enables the recovery of multi-scale features. And as it turns out, this is something that's going to be important for generalizing to different image properties. So going back to our diagram here of the different blocks, right? So the reason to abstract away the notion of neural network architectures into these different high-level blocks is that then we have these high-level knobs to tune. And specifically, we're going to be experimenting with this knob, the downsampling block, because we know that's going to increase the receptive field size and therefore enable us to better deal with the large instances, for example. So we know that there's a very, I mean, there's just a simple formula to describe how sets of repeated pooling and convolution layers can control the receptive field size. And as a visualization, you can see here that for convolutional kernels in an image, once you pool, now a single pixel after pooling has been influenced, has been computed from a wider or a larger area and the original layers before the pooling. So then if we increase the number of downsampling blocks, we increase the number of pixels that we can perform computation or extract features of as we go down in the encoder stack. So what do? We increase the receptive field size, and we vary the receptive field size and train networks at each of these different levels. And what we find is that for the fruit fly data set, which have tiny instances, although a large field of view and fine features, we do way better using the top-down approach than the bottom-up across the board. But that we also have a plateau in accuracy as we reach a particular level of receptive field size. And so what does this mean? It means that choosing even like the largest possible network that can integrate over a very large region of the image, especially when we're doing the top-down approach in which we first do the cropping, we can parameterize a neural network that is very specifically tuned to deal with the size of features and the data set that you that is specific to your particular data, right? And so as a point of reference, a UNet that has a receptive field size of 316 pixels and achieves effectively plateau level accuracy, effectively like perfect accuracy, has orders of magnitude fewer floating point operations than a general purpose ResNet 50, like that used in one of the deep loud cut backgrounds. And the reason for this is that rather than having a network that is a generalist, that is, it can handle features across many scales, across many different situations, instead create a network that is specifically designed to deal with the features of our specific data, right? It's about creating specialist networks as opposed to generalist ones. If we look at our accuracy in terms of space, you can see that we have virtually perfect accuracy, comparable to what we had in the first LEAP. In fact, even higher. Now the outer circles here correspond to the 95th percentile, so we can reduce the number of outliers considerably. And effectively, like the vast majority of body part predictions are within just 2 pixels of their ground truth location. So what about the other animals? Like say like our blobby mice, right? Here we find a little bit of a different story, where as we increase the receptive field size, we still see this improvement in accuracy, but we actually do considerably better using the bottom-up approach rather than top-down. And one of the reasons for this, we think, is particularly having to deal with the notion of having coarser features that, and therefore, and reasoning about spatial geometry at a global scale. So if you'll recall, in the bottom-up approach, the network sees the entire image at once, whereas in the top-down, we just see a centered crop, which when the animals are very large and encompass a large area of the crop, you're probably going to have most of the animals in that crop regardless. And so you have to the network now has to learn to reason about subtracting a lot of the background, ignoring a lot of the background, as well as other animals that may occupy the same regions of the crop that the focal animal would as well. And we see this again generalized to our B data set, in which we have these very large instances that occupy a very large fraction of the frame. And putting these guys side by side, you can see this trend once again of both the differences between the different kinds of approaches to doing multi-instance pose estimation, as well as the plateau in accuracy as a function of this architecture parameter of receptive field size. So this is all to say we have specific network architectures that do better for specific data sets, and the way that we can select those is guided entirely by the geometry and features of your particular data. So we can look at a couple examples, and then we can go to questions if anyone has any. So here you can see these might be a little choppy over Zoom, but you can check out the video link below. You can see on the left there a pair of interacting mice. You can see they're very closely interacting, in fact, in contact almost all the time, as well as the fruit flies, where some of them are even completely overlapping. We're able to keep track of all the body parts, including the ones that are slightly occluded, and their relative identities without mixing up body parts very frequently. As well as in the B data, in which we have many body parts that we're tracking in a very small region of the field of view. And yeah, cool. Okay, so I'll mention again that one particular observation about these different neural network architectures is that it generalizes both to the single animal case as well as the multi-animal case, right? And so the takeaway being that more carefully designed neural network architectures can perform better and with fewer operations and higher performance on when they're tuned to the data set that they're being applied to. Sage, is there any upper limit to how many animals can be tracked? There is not. The only difference is that in the top-down approach, the processing time is going to scale linearly with the number of animals. That is to say, if you have 2 animals versus 3, or rather versus 4, the 4-animal case will take approximately twice as long as the 2-animal case. Not quite the same, but at least for the second stage. But there is no hard limit other than memory constraints on your GPU, and honestly, it'll just run slower if anything. Jake asked about data augmentation used while training the networks for the results that presented, and does augmentation change results? It absolutely does. And so all these networks are trained from scratch without any pre-training, except for the ResNets, which we train both with and without pre-trained weights. And one thing that's crucial, especially when we're at the few shot, that is like we have very few examples, is to perform data augmentation. So this includes things like rotations, scale, and random, uniform, and Gaussian noise, as well as contrast adjustments. All of these have a huge impact in forcing the network to be able to generalize even within the same condition, even if it's kept relatively constant. So for example, even when we can adjust the scale to be fixed, as the animals are always the same distance relative to the camera, doing scale augmentation improves how much the networks can generalize because they learn to have more wiggle room in the image features that they capture. Remy asked, in the top-down approach, how do you deal with translation invariance of CNNs? Do you include position with respect to center in some way? So in the top-down approach, we do implicitly encode this, this position by doing this anchored crop. We can also do a bounding box crop, but in either way, the relative position of the body parts of the focal animal, the centered animal, are going to be with respect to the center coordinate of our crop. There's no explicit encoding like say you would have in like a 2D transformer or anything like that. The translation invariance is true up until a particular level of pooling. If once we do enough pooling and we have a receptive field size that is integrating over a large enough portion of the image, the kernel of a convolution deep in the network is actually not as translation technically equivariant as the initial layers. That is to say, its kernel is looking at all corners of the image simultaneously. And finally, would it also be able to differentiate between overlapping larvae? Yes, and again, this is all going to be like a continuously varying performance kind of situation, right? Where you're going to run into different structural sources of noise pertaining to like which body parts are present or not. We found that overlapping animals are a little bit more easily dealt with in the bottom-up approach than in the top-down. And you can think of this as relating to the fact that in top-down, you need to have a particular anchor point or centroid that's reliably visible in order to have this sort of a prior on the position of the animal within the crop. But your mileage may vary, and the best thing to do is just to try it out. Cool. I'm just going to move on in the interest of time and quickly tell you a couple of stories about how we've been applying these this sort of tracking to do unsupervised behavior recognition, and then we can move on to the tutorial. Okay, so as I mentioned, we consider our main way to quantify behavior in the form of movement is through these postural dynamics. And in particular, if we look at a set of stereotype behaviors, we can see that the dynamics, that is like these particular trajectories of the movement of body parts, has a particular pattern, right? You see like these oscillations occurring at regular frequencies, or in the case of grooming, a very characteristic shape that the movement of the body parts elicit. And so the goal then is to be able to recognize behaviors without actually giving them labels ahead of time by keying in on the statistics of the motion of the body parts. And the way we do this is by using an approach developed in one of the groups that I'm in here at Princeton called Motion Mapper, in which works roughly like this, in which say that we have several time series, and these could be like the motion of multiple body parts, in which we have characteristically different statistics of dynamics along the different segments of this clip. What we're going to do is first extract spectral features. So these are multi-time scale features. In other words, we're converting each of these time series into spectrograms. So this captures the power of these oscillations across different frequencies or time scales. And we're going to then concatenate all these spectrograms and slice through each time step, where now we have a feature vector that contains the power at different time scales for the different time series. And we're going to embed each of these time steps, this multi-time scale representation of each time step of our behavioral dynamics into a manifold, a low-dimensional manifold. And what this allows us to do is to capture the relationship or the structure across the different body parts in a lower dimensional space. And in that space, because we've pushed these points together, we're able to do something like density-based segmentation or clustering. And if we cluster in this space, we're going to have clusters of points in which we have stereotyped or very self-similar dynamics. And the idea is that self-similar dynamics should correspond to self-similar movements and therefore self-similar behaviors. So what does that look like? Okay, so in our single fly data set, an embedding will look a little bit like this. And great, we have a bunch of peaks and clusters that have kind of outlined, but no labels or no other way to infer what a behavior is from this directly. So the way that we're gonna be able to tell what's going on here is by assigning labels to each of these clusters that were previously identified. And we could do this in a variety of different ways. For instance, just looking at examples from each cluster and a posteriori giving these names to the behaviors. But I want to show you how using post tracking allows us to do this in a way that's a little bit more principled and gives us a little bit more information. So let's consider our map and look at specifically just one of these clusters right here. And so we're looking at this part of our behavioral manifolds. And what we can do is we can take all the points that fell into this region of the space and look at the average feature vector. That is, for every body part, what is the power at all the frequencies that we've measured. And what we can see is that for this particular cluster here, we have non-zero power at the wings and particularly of the hind legs at a wide range of different frequencies. So this can already lead us to believe, if we have some prior knowledge about how animals, how fruit flies move, that this is indeed going to be posterior grooming. And we can look at examples. So if we look at the adjacent cluster here, we find that this actually corresponds to hind grooming. And here you can see like the tracking overlaid on an example bout sampled from this state. And these are the relevant rows of that feature vector. So what you can see is that for the right wings, for the right hind legs, we have a high power, and for the left ones, we do not. Similarly, on the other side of this super cluster of behaviors, we have a left hind grooming, where the fly is moving its left leg primarily, and the average feature vector reflects that exactly. And our middle cluster here corresponds to our bilateral hind grooming. Again, you can see involves coordination of both of the legs. And they all share approximately the same set of frequencies at which those legs are moving, with lower power at the individual hind leg segments. So cool. This allows us to, without actually telling anything about our algorithm, like about what a fly is or what grooming behaviors are, identify purely from the statistics of how pose varies over time, these different kinds of behaviors, including ones that are mirror symmetric. But cool. So that allows us to capture one behavior that we couldn't before with classical tracking. But one that we could capture before is that of locomotion, right? So that was grooming. Here's what the feature vector might look like for locomotion. And at first glance, not terribly informative, because what we see is just power across all of the legs for some high frequency band. And great, we could have probably gotten something akin to this to say that a fly is locomoting just by computing the centroid velocity. But if we actually break down this supercluster along this little peninsula, what we find is that as we go from like the top region of this manifold here down to the bottom end of this peninsula, what's happening is that we go from like coarser, more diffuse frequency, leg oscillations to more sharply defined and at higher peaked frequencies to even higher peaked and more coordinated locomotion. And so what might not be obvious from the get-go is that if you look at the peak frequencies, this is the main characteristic that changes across these clusters. And if we characterize each of these clusters by their spectral distribution, we find that they are indeed characterized by these different leg oscillation peaks. So if we plot this against velocity, we find that there's a total overlap in the distribution of their forward velocities that is not present in the peakiness of their frequency tuning. And so they're somewhat continuous, something that we could not distinguish between before when we just looked at the forward centroid velocity. Now we have a very specific description of how that forward velocity is induced through how those legs are moving. So great, we've discovered some behaviors without any supervision, directly just from the posture dynamics. And so I'll move on to the future directions and tutorial. But before, I guess I'll just take a second for questions, and then we can quickly move on to doing this ourselves. So Matthias asks, how do watershed results compare to other supervised clustering methods like K-means or random forest? Can you keep state transition probability into account in order to do better segmentation of behaviors that never go together? Absolutely. So, I mean, this is a density-based clustering is sort of like the first pass, easiest thing that you can do. You don't need to use TZ or watershed. There's a good paper by Ben de Vivort on comparing across these different methods, including using things like Gaussian mixture models. But there's a whole litany of different approaches to doing supervised behavioral clustering, and including ones that particularly look at transition structure rather than being more agnostic to how the behaviors are grouped together. And I'm happy to talk more about that. We're writing a review paper currently that kind of goes through the general classes of approaches, but feel free to chat with me offline about these techniques. Cool. I'm just going to quickly move on to the future directions so that we can get started with the tutorial. Right. So moving forward, the things that we might want to do is to further help ameliorate the problem of having to do manual labeling of pose, particularly as it becomes more laborious with multiple animals. So there are advances in computer vision that enable unpaired pose estimation, where you just have a set of poses, and this can be perhaps derived from a different data set with images from your data set, and train a network that learns to disentangle the two such that you can produce pose estimation without doing any labeling at all, provided that you have some distribution of realistic poses for your particular kind of animal. Or using things like having a prior on the skeleton of your animal and allowing, and using fully unsupervised learning to be able to reconstruct, to force a network to generate confidence maps from which the original image can be reconstructed. This forces the network to learn to compress information about the anatomy without actually having to give it any labels. And finally, we're working on applying this stuff back to like our original model here of our simplified brain behavior generator through techniques for doing generative modeling, where we can use neural networks that actually output pose directly from sensory inputs. And this is a framework that can kind of subsume everything from supervised to unsupervised techniques. And some work has already demonstrated how you can use tracking to infer the sensory inputs of the animal, like projecting its scene onto its view, as well as using neural networks to output a pose directly, and then look at the neural network latents, that is the computations the neural network uses as a way to characterize those behaviors. All right, so I just want to do quick acknowledgements. Thanks everyone. Thanks a lot of the people at Princeton in particular who have helped and worked with me on this project, funding, collaborators, everybody at MIT. Thanks Janelle and Nat in particular for inviting me. And thanks you guys for listening. All right, and if we're ready, we can, I think, maybe move on to the tutorial. Yeah, so there it is. And we're gonna go through a couple of different steps here. And so as I mentioned, so if you go to the GitHub and you scroll down, you'll find the tutorial section. So you won't need anything for this part of the tutorial. All we're gonna do is we're gonna run some stuff in Colab, and I've already provided some of the data that we're gonna do this with. And so for the training part of the tutorial, we're actually gonna go through and do what I demonstrated in the middle part of our talk, which is to train a SLEAP model for top-down multi-instance prediction. So if we open up our first Colab here, we'll reconnect, great. If you click on that link, you'll see our notebook that looks a little bit like this. You wanna make sure that you're connected to Colab. If you hover your mouse over here, you wanna make sure that it says GPU as the Colab backend. And if not, you can go on runtime and change runtime type and make sure that the hardware accelerator is set to GPU. Great, we'll do that. And all that you need to do in order to run SLEAP on Colab is to run pip install SLEAP. Great, so we'll go ahead and say we're fearless adventurers, and we're gonna go ahead and click and install SLEAP. Should be relatively quickly. Should be done relatively quickly, and we'll move ahead as soon as that's done. And we'll be sure to be able to import SLEAP. So SLEAP will include everything that you need, including TensorFlow. And if you're doing this locally and you follow our conda instructions rather than pip, it'll also download the GPU drivers for you. But I highly recommend you try this out in Colab first. So while it's doing that, we'll start walking through the subsequent cells. So here we just have a few checks of the system. We'll come back to that in a moment. And particularly, we're gonna download our training data. And in here, what I've done is I've exported a couple of our training data sets. So this includes the images as well as the labels for our fruit fly data sets. And this is something that you can do for yourself from the GUI. So that later on, if you're trying this on your own data, you can just export a training package, as we call them. So these are single files that contain both the images and the labels, and get them into Colab by just downloading them, like using curl, or Colab also lets you just click here on the side and just upload files directly, or mount them through your Google Drive. But for now, we're just going to download them from my Dropbox because it's easier to do it in a single line. So let's check here how our installation is going. Almost there. uninstall some things in addition to installing. Cool. Maybe if anybody has any questions, now is a good time to try that out.
So I have a question, Talmo. If we want to run it not from Colab, but from our own computer, can you show us how to do it?
Yeah, absolutely. And so at the bottom of our tutorial here, we're gonna have a section that says, how do I get some more sleep? Something in short supply for us grad students. So you can go on our website at sleep.ai and follow the tutorial for both installation, creating a project, labeling, and we have everything step by step for all your OSs, including little animations of how to go about adding videos, defining which points you want to track, to generating your initial labels, training, and predicting them again. Going through the whole human in the loop procedure, as well as then exporting your data for analysis afterwards. So our website's pretty complete with the tutorial that I'll walk you through. It's just a little bit more, I guess, worked out. So, all right, we've done our pip install here. Everything is working. We've run the cell here. And it shows that here we have our GPU, and Sleep is able to detect that the GPU is available and accessible. So we'll download the training data. Should be relatively fast, but take a couple of seconds. We'll unzip it, a moment, and then we're going to configure our training job. And so this section down here, yeah, there you go, we've downloaded our training files. In this section here, we're going to define a sleep training job. So as we covered in the talk, there are several different approaches and many different neural network architectures that you're able to use through Sleep. So we've created this configuration system that allows you to basically specify every aspect of how the data is handled, to the neural network architectures, to the outputs that you want the network to generate, to how you want the training to be done, as well as what metadata you want to generate. So here, this is just a configuration, so we'll just run the cell. We're starting off from our default job configuration. That's all these different sections. We're going to just edit a couple of the parameters so that we can do our centroid model prediction to then set up our training job. And at this point, the Sleep trainer will load up the videos, detect all aspects of what size are your images, what format, how many parameters, create your network, there were parameters and layers, hook it up to the correct outputs, load up your different training sets and optimizers, as well as creating visualization hooks. And so because we're in Colab, we can also run TensorBoard directly in our browser. Cool. And you'll note, by the way, that Sleep already created a models folder here in which our specific run, the baseline model for centroids, is going to be stored right here. And you'll note that amongst these files, we also have these JSON files that contain a serialized representation of those configurations so that you can always reproduce it, as well as a copy of the training data. So this allows you to fully reproduce a model from every run. But cool. We have our TensorBoard running now, and all we need to do to actually get Sleep to run is call our trainer.train. So this will actually start generating the training data and feeding it into our optimization loop. As soon as it finishes preloading the data, you'll start to see how it will do everything from loading, formatting, augmenting the data, and then feeding it into the network for training. So great, generate the data sets, we're starting the training loop, and we're gonna do it for 200 batches for 50 outputs at the most. So already we're streaming some stuff into TensorBoard here, and the first thing you'll see is that in this graphs tab, you can visualize the specific architecture that we configured. And so this is our UNet architecture. We start from here. We have a series of convolutions and pooling. So just like that diagram I showed you before, we have pooling steps and skip connections that go up and connect the encoder to the decoder. Cool, that's our architecture. But if we click on scalars, we'll begin to see the outputs of every epoch with respect to the training and validation losses. And a little bit more informative is if we click on their images, you'll be able to see a visualization directly of the actual predictions of the network so far. So if we click on this, we see a prediction on the training set and on the validation set. And as you can see, even as of the first epoch, it's already doing a reasonable job of predicting these confidence maps, these big blobs here, where the red points are the predicted location of the anchor points, of the centroids, and the green ones are the ground truth locations. So even with a single training epoch, we're already pretty close. That took about 30 to 20 seconds. You can see that it goes by pretty quickly. Every iteration, right? And so we'll leave that guy training for now, and later we'll be able to scroll through these guys and see how the training progresses in real time. But after we finish the training, we'll have this best model, the best model so far, in our models folder, and we'll be able to download it locally with this last cell here, which will run after that's finished training. In the meantime, we can already start in parallel our second model training. So this is going to be our top-down confidence maps. This guy is going to be a model that takes as input those cropped images and predict the confidence maps for the centered animal. So I'll go back to the tutorial page here for a second and show you that this is what the final predictions will look like for that interactive training of the centroids. And after we do our interactive training for the top-down model, we'll get predictions that look a little bit like this for our centered animals. And we'll look at that in a second while it's starting. After you finish running both of those, you're going to have downloaded 2 different zip files that will contain the actual trained models. You can run those locally, or you can upload them to Colab to do inference or predictions on new data. If you want to run either of these notebooks locally, you can just clone the repository and download the data using these commands. So you'll have a folder here for your models, as well as for the training data. They're kind of too big for GitHub, but the links are there if you want to be able to reproduce this locally. So let's check in on our training. Here's our centroids. We're at the 10th epoch already, and we can look at how the training is going on TensorBoard. Refresh here, and it seems like the network's already converged to be able to produce these confidence maps very reliably. Great. Both on the training and on the validation set, which is held out. Cool. So in parallel, we're also training in a new tab here. Installed SLEAP, we're still installing SLEAP here. We'll soon then be able to start the training on the top down. But maybe in the interest of time, and we can hop back here in a moment once this guy is finished running. And by the way, these notebooks will take about 30 minutes from end to end to install, get the data downloaded, train, and download the results. Cool. And this one is formatted much the same way, and the only difference in this second notebook is that we're going to be training our top-down model, so our configuration is going to be slightly different, where now we have an output head of our model that outputs the centered instance confidence maps rather than the centroids. And we'll see what that looks like in just a couple of moments. It's like we're just downloading the data here. Cool.
While it's downloading, can I ask a quick question?
Yeah, absolutely.
So what are the sort of major failure cases that you might run into if you're training this on your own set of data? So for instance, like are there any tricks with initialization that you need to like be aware of with the networks?
Yeah, absolutely not. So in fact, like all these networks are trained from scratch. And so we put a lot of work into making it seem as magic as possible. But definitely, as you saw from our from the presentation, different approaches will perform better on different data sets. And it's not exactly knowable a priori which thing is going to be the most optimal for your data, but we do provide these sorts of points of reference. And so when you're training this through the GUI directly, we have some baseline job configuration profiles that are that tend to work in the general case, but maybe won't work the best in your specific case. We'll see how the GUI looks like in just a little bit. But for the most part, all you really need to do is do the labeling, do it for 10, 20 frames, and just try out training. I usually like to start off with the top-down approach because it's a little bit more flexible because you're breaking up the two different components, different networks, and see how well you're doing at that point. It'll import the predictions back in, and you'll get a baseline sense of how it's going. You'll also be able to visualize the training in real time, even when you're doing it through the GUI. That'll immediately give you a sense for whether there's a major failure mode and how you've configured SLEAP. We have a lot more information in the website as well. Clay asked, could SLEAP also work well for free-moving mice installed with a miniscope and a wire hooked on their heads? Yeah, absolutely, and we've done that before. I don't have that immediately available here, but yeah, you can, if you want to track the implant, the miniscope, for example, you can define that as an additional body part to track, or you can just choose to ignore it altogether. And either way, it'll be able to handle it. So what people have done in the past, using either LEAP or Deep Lab Cut to deal with multiple animals, is to treat the separate animals as actually the same instance. That is, you'll create, you know, a bunch of body parts that are called, like, you know, a forehand or the foreleg of animal with implant and animal without implant, or the white mouse and black mouse, right? Something that distinguishes their appearance. And then the network will hopefully learn from context that even though individual body parts may appear very similar, that looking at, you know, taking into context a particular feature that differentiates the animals will allow it to predict the different, to assign the body parts the correct, you know, pseudo name or label. This is not ideal because if body parts appear similar, what you're going to see is a flickering of the prediction of that body part between the two different animals. Because you're not really explicitly representing the notion that they're different instances. SLEAP handles all this transparently. We actually treat each animal instance as if, like, you have multiple copies of the same body parts. So if there is a body part or landmark that is unique to a particular animal, that's fine. You can add it to the skeleton and basically mark it as not visible in the other animal. And it should just handle it transparently. Cool. All right, so because we're almost out of time, let's just have a look at how we're doing here. So here's our centered instance confidence map network. Let's switch over to the images. And as you can see, in the time it took me to say that, whereas we start with like relatively rough predictions of where the individual body parts are, very quickly, over the course of just a few iterations, now we can very quickly converge on to being able to detect all of those different features. And it still can get some stuff mixed up. As you can see here, it'll maybe still assign some body parts incorrectly, but it'll very quickly learn to resolve those. You can see there. And again, the training will keep going, just like it did over here, and eventually you'll be able to download the trained models. So in lieu of actually waiting and downloading those, I've already trained those and made them available so that you can just download them. And we can move on to like how we're going to predict on new data. So this is going to be a third collab in which we're going to take an unlabeled clip and apply these models to generate new predictions. So let's connect to collab again in this third notebook. And all right, I'm going to have to wait a second so that it can install SLEAP again. Every time this is one of the downsides of using collab, is that it does have to install everything from scratch every time you start a new instance, but we'll live with it. That'll only take a couple of seconds, a couple of minutes rather. Maybe I'll take this time to also demonstrate what's going to happen next, which is after we run the predictions, we're going to get the output in a single file, then we can download. But we can then visualize the predictions locally. I'll show you how to visualize it on Colab, but most of the time, you're going to be running the predictions either, doing the inference either on the cluster or on Colab, and then downloading it so you can actually look at the results on your own computer. And for that, you can you can look at them through the SLEAP GUI. I have some instructions here on how to install SLEAP in a new Conda environment, which is the easiest way to go about doing it. And then here I have a terminal already open, which I'm going to activate my SLEAP environment. And then I'm going to call our SLEAP label shortcut on the prediction. And I've already saved the predictions file, which we're going to see how to generate in just a moment once Colab finishes installing over here. But this is what the final result will look like in our GUI. And so here we've opened our predictions at SLEAP, and we can scroll through all the frames of the video, as well as say, zoom in and be able to inspect the results a little bit more closely. And so you can do that just by scrolling your mouse wheel. And we have all sorts of interesting information here, like the tracks that the animals were assigned to. This is like the skeleton. Here's a video that we use for prediction. And you can see that it not only is able to keep track of the animals contiguously, but the key points are stable, they're accurate, and it's able to handle even when they're very closely interacting. And again, this is on unlabeled, held out data that was not used to train those models. So we can go frame by frame, and you can see how SLEAP handles situations where you have things like motion blur, for example, as well as heavy occlusion. So with these results, so in the SLEAP GUI, you could, if we had any tracking errors, you would have been able to correct them here by transposing the tracks, deleting them, or otherwise correcting the predictions. If you want to use this for training, you can just double-click one of the instances, and that'll create a training instance from our data. We'll do this for, say, both of these guys. Mark all the points as there, and we can say click and drag to just correct those if we needed to. Typically, I'll do this in a single labeling data set that contains more than a single video. That's what our GUI looks like. And once you're happy with it, you can export an analysis file that you can load up in MATLAB or Python. Cool. Oh, looks like we're done. All right, so here, what happened in our inference notebook is that we downloaded the models that we trained using the other two notebooks, and we downloaded little test clips, an MP4 file. The way that we do inference in SLEAP, if we're doing it interactively, we have a command line interface as well as the ability to do it through the GUI directly. In the GUI, you can just go to predict and run inference and select your models. But on Colab, we can create it interactively by importing our predictor classes. So we can create a predictor, a top-down model predictor, by giving it the path to our two different models that we trained. Then create a tracker that associates those detections across frames. SLEAP has support for a lot of different types of video data, and we can, for the most part, just load them in directly just by doing sleep.video and load them from the file name. It'll figure out everything from its format, as well as treating it effectively like a NumPy array, such that video I/O is relatively transparent. And this allows you to create custom pipelines if you want to integrate it into your some other analysis or processing pipeline that you already have set up for your data. Okay, cool. We have data loaded. We have the predictor set up, and from already have loaded our trained neural network models. Then we can just predict on the video, and we're going to set the make labels equal to true so that we can create one of these labels files. That's the thing that we loaded up into the GUI. And that is give us our predicted labels object. And great. Once we have that guy, we can plot them. We have some plotting utilities, and here I just set up some interactive plotting in Matplotlib, and you can see in Colab. So you can see how we can scroll through different frames, and in real time we can, in Colab, load up the frame, load up the predictions, and overlay them. So as you can see, like this is, obviously not as interactive or as high res as you can get with the GUI, but it's a useful way to be able to inspect the results of SLEAP directly in your Colab notebook. And so obviously, if you want to do it in the GUI, what you'll want to do is save those labels out into our predictions.sleep file, and then download them directly from Colab, which you could also have done over through the files tab here in Colab. Ta-da. Great. So recapping, what we did was trained some centroid models that could predict the location of anchor parts of multiple animals. We've trained a top-down part detection model that can, given a crop of a centered animal, predict the location of body parts. And we loaded up these trained models such that we can now track a new contiguous clip of new data, and downloaded the results so that we can inspect them locally on our computer. And you can do this, say, on your laptop or something without actually having access to a GPU. And that about covers it. So thank you guys. If you're interested in using SLEAP for your work, check out the link at the bottom here. Give us a Twitter follow, and we'll be announcing new versions of SLEAP with new features for more fancy tracking and getting our quantitative behavior on. So thanks, guys.